And we now move over to our next speaker. You might have recognized that we kept the time slots quite short to keep this afternoon lively and open for new ideas and discussions. Such a new idea is now brought in by Merlin Lindemann. Merlin is holding a PhD degree in animal sciences from the University of Minnesota. Currently, you are holding the rank of a professor at the University of Kentucky. And just recently, you received the Cromwell Mineral Nutrition Research Award. Congratulations. And you were named as a fellow of the American Society of Animal Science. Merlin is going to present, could you click already to your slide, please? Is going to present the topic sustainable swine production through an improved understanding of feedstuffs and the animal. Merlin, please.
Thank you very much, Franz. Last year in November, I had the privilege of being down in San Antonio at Biomen's American Nutrition Conference, and I enjoyed it so very much that when they asked if I'd be willing to make some comments here, I readily accepted. I think it's an excellent forum that you put together for the industry. As he said, he wanted to keep these things short. I've got quite a few slides, so if you've got a seat belt there, you might want to put that on, or at least grab the handles of your seat. So we're going to roll through some things. The previous speaker gave us some excellent ideas about things we can do currently that might improve production and potentially improve profitability. We have those technologies already. What I'm going to talk about is some things primarily that look 10, 15, 20 years down the road as some of the different changes that might come about and the technologies that you may be using in the future. The reason to do that is to give you hope that there will be a continued advancement in knowledge in order to meet the needs of a growing population. We see these slides all the time about an increasing population. People argue or debate whether it's 8 to 10 million people, but it's going to be growing. But we see that growth is not throughout the whole world, not in the industrial countries, but it's primarily in the developing countries. Now, associated with that growth in number of people is an increase in economic buying power. But can we feed all those people? You know, a pessimist sees a problem, an optimist sees an opportunity in that. If we look at some of the developing areas and we look at projections from 1970 on through 2050 of what kind of increase in food consumption there'll be, one of the things that stands out is this huge increase in meat consumption. As buying power increases, there's a lot of things that are key interests for the consumer. One of them is technology goods, so iPhones, iPads, things like that. But meat in the diet, animal protein, is a key thing that people want to consume. And if you look back at the consumption, this is the worldwide, but it holds for other segments of the world too. The largest meat consumed is pork. There's a fair amount of poultry, a lot of beef, but pork is number one. So we've got a growing population that desires pork. So if we can produce the pork, the market's going to be there for it. Now the question is, how and where do we produce the pork, and how will we feed the pigs? We had a little bit of talk in the first session about whether because of population pressures and legislative pressures, pork production may shift somewhere. But it will be produced, and how will we feed those pigs? Because we need more cereals too. If we're going to produce more pork, we need more inputs, or we're going to have to use the inputs we have more efficiently. Now, personally, I think there's very limited potential for improvements in these things. I don't think doing another lysine titration study to see if we need 0.02% more digestible lysine for the pig is going to change things very much. We'll have some improvements, but we have worked those areas pretty hard. I think that the things that are going to come about are new things. We're going to have to expand the box of knowledge, not just improve the knowledge that's inside it. And so there's areas of gut microbiology and mycotoxin control, bioactive peptides, a variety of things that new information is going to give us new technologies that we'll incorporate into our diets. Some of these things may be for specific nutritional needs. Other parts of them maybe the additives that help us to use those things more efficiently. Now, talking about gut microbiology, the subject of this session here, we know there's been ban of antibiotics in some markets. We talked about that in the first session. It's always talked about. But with regard to intestinal microbiology, there are new procedures that we have, technologies that have come about in the last 10 years that allow us to identify and type the bacteria that we never could before. When I was in college, you had little Petri dishes, little plates, and you swiped something on there and you talked about 6 or 7 different classes of bacteria. Now we are able to identify things that we never could before. The only reason I have this is to say, here's some of the new information we get, and I don't have the first idea of what that says. It's that new, and that's not a part of my training. So if you're in a major company, you need people, young scientists who are trained in microbiology. All these types of bacteria listed there, those were names we didn't even have in front of us 10 years ago. Now, we can also talk about in the human area, there are actually fecal transplants going on, putting bacteria from a healthy person into an unhealthy person to cure various types of diarrhea and for diseases like Crohn's disease. This goes on all the time. But it's current, and recent. It's within the last 3 to 5 years. There's talk about whether a baby is born naturally versus cesarean birth, whether that changes the gut microbiology on the baby. And it does. But just this past Sunday, I read something that placenta bacteria is now being characterized, and we realize that the developing fetus is getting bacterial profiling from the mother through the placenta. And there's thought that microbiota composition may be linked to early infant growth rate. Very current. Now, you can just translate that. If we use pigs as a model for humans, we can use humans as a model for pigs. We can use that information. So there's a lot of information about this gut microbiota established during infancy that contributes to obesity, and let's just say energy yield later in life. So it's energy yield that we can capitalize on in the pig area. And so being a scientist, I know that sometimes we administer antibiotics that way, but I'm a thinker too, so there may be some new way we can do this thing. So all we got to do is turn that pig around, that pig, I don't know, he may not like it, but hey, there's possibilities. Now, if you go to Mayo Clinic, and it's a well-known worldwide clinic in Minnesota, they've got places throughout the U.S., but the clinic in Arizona does a lot of fecal transplants, and you have your choice. You can get administered through the nose to the stomach, through the nose to the intestine. You can do through the mouth an upper tract endoscopy into the intestine. You can do a colonoscopy if you don't like the facial route. You can go the other route. You can do a retention enema. There's a variety of possibilities here. Now, most people that are on the farm don't like to do things to go somewhere else. They'll do it themselves. Do-it-yourself bookshelves or greenhouse if you're a carpenter. You can do a do-it-yourself computer. The plans are everywhere. Or do-it-yourself plumbing projects. Well, you can go to the internet, and they've got fecal transplant do-it-yourself instructions for you. They'll put you a supply list, tell you how to do it. There's even YouTube videos on this. So what I'm telling you is this technology is coming. So let's talk about gut microbiology a little bit. Recent papers, but gut microbiology affects neurotransmitters. It affects, vagal control, that's going to affect motility in the gut and other things. So many things that microbes affect. Just to look at a couple of these, we can take the microbes from an obese mouse, put them in a mouse that was not obese, and what we see is that we have a greater energy yield. We get an energy yield increase in acetate and butyrate production. So if you look at then the energy in the feces, the energies of the feces of that mouse that got that gut microbiota from the obese mouse, there's less energy. More has been taken out of it. And as a result, that normal mouse puts on more body fat. just because of changing the microbiota. And because it's fermentation, that's an energy yield from the non-starch polysaccharides that aren't digested in the small intestine. So there's opportunity to capitalize on this in the future, and I think this is one place. Understanding the gut microbiota is going to enable us to use lower quality feedstuffs and get more energy out of them. This is just another thing to show that obese mice, right here, have a different bacterial profile than the lean mice. So the bacterial diversity in the obese mice is less. And they do a lot of things looking at similarity between different classes of people. But the microbiota in lean and obese mice is different. And we may have been selecting to some degree for the microbiota all this time that we thought we were making genetic selections. It was a microbiota selection. Here's an interesting one. Again, very recent publication. You can take some germ-free animals. You can put an obese mouse on a high-fat diet, and that obese mouse becomes fat. So that's what always happens. You can take a germ-free mouse where has no microbiota, put it on the same diet, it doesn't become fat. So again, part of the reason that obese mouse becomes fat is the particular microbiota and how that microbiota interacts with the diet. Now here's another interesting one. You can take germ-free mice, put in the microbiota from normal mice, and you get a normal mouse growth. Put in the microbiota from an obese mouse, and you get a fat mouse. You put in the microbiota from an underweight or very lean mouse, and that mouse will lose weight. All because of the microbiota that you have inoculated the gut with. So this is a place where there will be advances in the next few years, either by adding things such as essential oils or phytonutrients that may control the gut microbiota, or by specific technologies that allow us to inoculate microbiota. There's been studies looking at inoculating pigs with human microbiota, and so this interspecies transplantation is going on already. And at our university, the ruminant nutritionists have been talking with me about starting to get some work done with transplant of rumen bacteria into pigs. We'll see if it happens. I don't know. But microbiota affecting brain behavior, immune system, nervous system, all of these things already known in the literature. Now, because I am a nutritionist, I'll say a few things about nutrition, but not a whole lot. But I really think we need to look at diet formulation as going beyond the nutrient requirements to being a part of risk management. The least cost diet does not equal the greatest potential for profitability. You have to make your decisions based on ROI, return on investment, for anything into or out of the diet. The lowest cost diet does not optimize production flow. And I'll give you a very simple example. Here's work, small difference in price. That's a cheaper diet than that. Look at the difference of the pigs, obviously. Well, that's a deficient diet with zinc, but I think it illustrates the point. You can't always go for the least cost. Now, pigs are different. We saw some pictures earlier of some Chinese breeds, but there's a lot of different pigs out there. Some are good reproductive, some are good for growth rate, variety of things. Our modern pigs, there's differences out there. We can just go to this example. You take those two vehicles. If you go up to a pump, you got choices. You can put the cheapest gas at the pump in this old car. She'll probably run all right, pretty good. You put even the best grade gas in this, and you aren't going to win the race. If you got modern genetics and think you're going to get out of them what the geneticists have put into them by least cost formulation, you're not going to be here next year. You know, it's not something that works. And here's why I bring this up. I've done work with mycotoxins, aflatoxins in the 80s, fumonisin in the last 4 or 5 years. If you look at a summary of a variety of studies, and you look and you see there's going to be a decrease in growth rate, greater decrease with more toxins, whether it be aflatoxin, fumonisin, or DON. Same thing. And there's a variety of products on the market that you can include. We know, just from BioMin's publications, these things are around the world. They've always been here. They're here every year, depending on the weather, depending on the soil type. Every single country has them. The thing is, we've got variety of incidents of mycotoxins, and if we look at the levels, we can talk about a level of 29 part per billion aflatoxin for these 1,000 samples. The high level was 2,200. The point here is, if you're purchasing corn, how do you know where it was grown, what the growing conditions were? How do you know how the mycotoxins have changed during transport and shipping of that to your country? You know, it's very different. So you need to have a mycotoxin in there, binder in there. And here's one of the reasons we had climate change this morning that we talked about. Here's for the U.S. with a very low emission scenario of carbon or a high emission scenario. That's how the temperatures are going to change in the U.S. over the next about 80 years. Now, we can put this scenario in there, the state of Michigan, in a low emission scenario, it'll get this hotter. And by 2080 or 90, we're down to here. And a high emission scenario, this is the climate that'll be more typical. The reason I put this up is here's where the corn is grown for a lot of you who are purchasing it, and in another 60 to 80 years, the climate will be more like this. If that's the case, we don't have the water to do it. Things will change. All right, we'll just mention one thing. Beyond bioactive peptides, it'll be another thing that comes on. We're going to be putting things in that we don't look just at the amino acid profile, we look at the peptide sequence. 70% of the amino acids that your pigs absorb are absorbed as peptides and not as single amino acids. The digestion does not take it all the way to individual amino acids. All right, we'll just say this, that here's an example of products already out there, a fermented product. What happens if we feed this fermented potato protein? The IGF-1 levels increase in all pigs, and especially in these low birth weight pigs, they go up. Those pigs have an advantage. So these bioactive peptides are going to do something other than just supply amino acids. They're going to stimulate certain parts of the body. So we're going to skip that. We're just going to say that a growing population desires pork. One person's problems are another person's opportunities, and the new technologies of many types are going to give us hope for success and profit in the future. So thank you very much.
Thank you very much, Merlin Lindemann. I was wondering whether this piglet was volunteering for a second shot. A quick question to Merlin. A lot of topics mentioned there. We still need to digest all this information. If this is not the case, we proceed with our next speaker and we keep our questions to Merlin to the final Q&A session.